So the first talk, sitting home, and kind of like exploring patterns of growth and drought resistance in tepary bean.
All right, so our species is tepary bean or Phaseolus acutifolius. And this summer I have been working on your documentary pattern. So, in order to understand the importance of the species, we have to understand its habitat. So the tepary bean is native to the Sonoran Desert, which ranges from the southwest portion of the United States down to the northwest portion of Mexico. So this region is characterized by being hot and dry. It's a desert, but the landforms of the region form unique weather patterns. So specifically what will happen is you'll have a cloud of precipitation go up and over the mountaintop, and once it crosses the peak, then we'll have like a hard precipitation event. And so all this water will flow down the side of the mountain in a channel of water called a desert wash, and then it'll flow out once it reaches the bottom in a delta called a alluvial fan. So between the desert wash and the alluvial fan, we have the perfect environment for water retention and nutrient retention, which makes it the ideal location for germination for a lot of desert adapted species. And then another point of interest lies in the soil composition of the region. So a lot of people think desert and they think sand, but this actually isn't always the case. So a 2018 soil survey done by the U.S. Army Corps of Engineers, they sampled from Tucson all the way to Phoenix in the Sonoran Desert, and they created a soil profile. So on the soil triangle here, we are looking at the orange rectangle, and as you can see, the soil composition for the alluvial fans range from 80% sand all the way to 80% silt. So the difference in particle sizes creates differences in pore sizes in the soil, and pore spaces in the soil contribute to water retention as well as soil aeration. So we're interested in looking at the soil aspects of that. So tepary bean is extremely drought and heat tolerant. So studies have been done to identify the specific mechanisms and genes behind these traits to introduce them to the commonly cultivated bean, Phaseolus vulgaris. So the tepary bean was and still is used by indigenous tribes in the region, specifically the Tacoma people. And it's perfect for this because it has a short lifespan with rapid reproduction, as well as producing highly nutritious beans despite a short reproductive cycle. And then current studies are very limited on the species, and the ones that do exist focus on the later life stages, where we're interested in looking at germination in Supernova. In the Sonoran Desert, the tepary bean coexists with the creosote bush, or Laria tridentata. The creosote bush has the nickname greasewood because it produces an abundance of bioactive chemicals, which includes a large composition of terpenes. So the creosote bush releases these compounds into the air and into the soil, and due to these two species' close proximity to each other, it stands to reason that the roots of the tepary bean would be interacting with the terpenes from the creosote bush. So previous studies in the McKenna lab have looked at thymol, which is a specific terpene, and its impacts on other legumes. And they have found that thymol stimulates modulation, growth, and development in other legumes. So we wanted to carry on with the study of thymol and see if they have the same effect on tepary bean. A 2025 study, so this year, they linked thymol to the activation of a gene that controls lateral root formation. And this builds upon a previous study in 2019 that identified that lateral roots use the same genetic pathway as inoculation. So essentially, if you can use thymol to stimulate lateral root development, you're encouraging nodulation at the same time. And this is super important for the tepary bean because the Virginia State University study found that on average, tepary beans only have about 2 nodules, and they're located right at the top of the root system. So legumes rely heavily on nitrogen fixation to complete the life cycle. The ability to have robust nodules given their environment is incredibly important for this system. So, our first experiment, we wanted to look at the soil aeration, effect germination, and growth in tepary bean. There's a novel method coming out called air pruning, and so what air pruning does is it increases aeration of the root zone by allowing more holes at the bottom of the pot. You can see that here. So a standard pot would have 1 hole, where this one you can see has 5 large holes at the bottom. This just allows more air to reach the roots. So to accomplish this, we set up 2 trays of 49 cones. One of the trays had wire mesh at the bottom to allow for increased aeration, while the other tray had potting grounds at the bottom to limit the aeration. We used a 2-to-1 sand to soil mix to imitate the soils of the Sahara Desert, and then we inoculated the seeds with the seed Phaseolus mix for modulation. We kept these seedlings in bay 1, and then we had temperature problems, so we had to move to bay 3 and bay 3. We watered them as needed, we rotated them for appropriate sunlight, and we tracked germination data. And then at the end of the during this process, we also monitored root expansion, or the number of days it took for the roots to reach the bottom of the cone to visibly stick out. At the end of this 10-day period, we moved on to harvest and processing, and this was done by separating the shoots from the roots, drying them, and then weighing them for comparison. So we found that shoots emerge faster with higher aeration. On the left here, on the y-axis, we have days since planting, and on our x-axis, we have our treatments, low aeration and high aeration. And as you can see, the high aeration group had a shorter time between planting and shoot emergence than the low aeration group. Similarly, roots expand faster with high aeration. So again, we have days since planting on the y-axis, our treatments, low aeration and high aeration on the x-axis. And again, you can see that the high aeration group had a shorter time between planting and root expansion than the low aeration treatment. So it was the same. Well, limited studies exist on germination and early development of plant species, and we found that soil aeration speeds up shoot emergence and root expansion. So given that tepary bean likely germinates in the desert wash and alluvial fan area, what are the impacts of the intermittent flooding here on early germination? More data is needed on our species. Our second experiment introduced the terpenes. We wanted to know, does thymol or other compounds exuded by malaria affect shoot or root development of tepary bean seedlings? To accomplish this, we set up 3 trays of 49 cones each, all with cotton nuts at the bottom. We had 3 soil treatments: the control, which received no additions, the thymol treatment, and malaria treatment. Malaria treatment was created by using dried leaves of malaria bush, hickory soap bush, and it was done through a heated leaves extraction, so a tea essentially. And then the thymol solution was created through a serial dilution of stock solution. This time we used 1-to-1 sand to soil mix, and we did not see an ovulation in our first experiment. So to encourage this, we used the same formulation, but with an added element to help with sticking to the seeds and hope to see ovulations. Again, we watered as needed, recorded germination data, and this time we measured leaf chlorophyll, recorded stem plants as well. We harvested after the same 10-day period, and then dried and weighed the shoots for roots for comparison. What we found was that thymol and malaria extract showed different patterns for root rate. So, for shoot rate, my apologies. So on the y-axis here, we have average shoot rate, and on the x-axis, we have our treatments, control malaria and thymol. And as you can see, the shoot rate in malaria and thymol did not differ from the control. However, the thymol treatment had significantly greater average shoot rate than malaria. Likewise, thymol and malaria extract showed different patterns for root rate. Again, on our y-axis, we have average root rate, and on our x-axis, we have our treatments, control malaria and thymol. And you can see that the malaria treatment had an average root rate, but that was significantly greater than control. And then finally, looking at chlorophyll concentrations, we found different patterns with thymol and malaria as well. We have the chlorophyll concentration on the y-axis, our treatments on the x-axis again, control malaria and thymol. And you can see that the thymol treatment had significantly greater leaf chlorophyll content than the control. So the big idea here is that thymol and malaria extract use different effects on tepary beans. Thymol appears to have a positive effect on photosynthesis, which is indicated by its significant increase in chlorophyll content, whereas malaria was demonstrated to have a positive effect on root biomass. So thymol is an isolated terpene from malaria. We only use one concentration. So future research could look into the effect of different concentrations of thymol on seedling growth, as well as the different compounds in malaria have been identified, but not their concentrations. So the individual effects of each compound and their concentrations based off the extraction method could also be tested. And then our main goal here is to inform ecological and evolutionary understanding of desert adapted plants. I'm happy to say. Yes?
Did you look at nodulation of the roots?
We did look at nodulation. So we allowed ours to grow for a period of 10 days. And again, there's not a lot of existing information on the species. So we found very limited number of nodules. And this makes sense given the Virginia State University study that they studied different varieties of tepary bean, and they found anywhere from 0 to 3 nodules on average on theirs. So the fact that we didn't see it could be due to a variety of factors, but isn't necessarily unusual. But we did this. Yes.
Did you come up with any good ecological ideas for why it might be better to invest in roots in the presence of Phaseolus?
To invest in the development of the roots?
Yeah.
Yeah. So tepary bean is a wild bean. It's also a crop. So a lot of you would think that it's very important for fruiting and to emphasize the shoot development. However, with the root development, you're in a desert environment. There's going to be phases where there's severe droughts. And you need to have a robust root system in order to survive these drought events. Thank you so much.